The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
All right, so today we're going to be talking about environmental risk factors for autism, and particularly the evidence suggesting that organophosphorus pesticides may contribute to autism risk. Before I get into talking about the OP specifically, I want to first review for you some of the evidence that really suggests environmental factors are important in contributing to the risk for autism. Some of the most compelling evidence that environmental factors contribute to autism risk really come from studies of the prevalence of autism over the past 20 years. And so that's illustrated in this slide here. Panel A on the left shows you the incidence of infectious diseases between the years of 1950 and 2000. And you can see the rates for these infectious diseases have dramatically dropped. This is because of the advent and widespread use of antibiotics. However, in contrast, if you look at the slide presented, or the graph presented in panel B, this is looking at the incidence of immune disorders over the same time period, and you can see that there's been a dramatic increase in diseases with a predominant inflammatory component such as multiple sclerosis, Crohn's disease, type 1 diabetes, and asthma. If you look at the red line, which is autism, that has been superimposed on this graph, you can see that the increase in the incidence of autism has outstripped all of these other immune disorders. These data basically suggest very strongly that environmental factor is important in this increase in the incidence and prevalence of autism, that the increase has occurred much too rapidly to be explained on the basis of genetics alone. Now, one of the questions that's been brought forth in regards to this type of data is how much of the increased prevalence of ASD represents an actual growth in numbers. Could it be that the increase that we're seeing really reflects increased awareness of the disorder, improved detection, and/or broadening of the diagnostic criteria? So in other words, is this concept of diagnostic substitution. People are being labeled as autistic who previously would have been diagnosed with something else or not as being autistic. This question has been evaluated scientifically. There are a number of reports out there now that have all come to the same conclusion as the study that's presented on this slide. This is work from Dr. Hertz-Picciotto, a colleague here at UC Davis, and her colleagues, in which they looked at the increase in cases in California from between the years of 1990 and 2006. And they asked, what is the increase in cases, and what are the potential causes for this increase? And so you can see their data suggested a 600% increase in cases across this time frame. Of these, 25% could be explained as earlier diagnosis. 56% appeared to be attributable to inclusion of milder cases. In other words, cases of autism that previously would not have been diagnosed as autism. And then 120% of the increase is likely due to changes in diagnostic criteria. But the important take-home point here is that even though diagnostic drift or diagnostic redistribution can explain some of the increase, 400% of the increased cases cannot be attributed to diagnostic distribution, suggesting that other factors, potentially environmental factors, are contributing to a real increase in the number of autism cases. So is there additional evidence besides this increased prevalence? And actually some of the strongest evidence implicating environmental factors and increased risk for autism come from genetic studies. So provided here is a list of some of this evidence. For example, incomplete monozygotic concordance. What does this mean? So basically, monozygotic refers to twin studies, and these are identical twins that share the same genetic background. If you look at autism in these monozygotic or identical twins, there are many cases where the one twin is diagnosed with autism and the other doesn't, or that the phenotypic manifestation of autism is dramatically different between the identical twins. If autism were strictly a genetic disease, you would expect near complete concordance or agreement in twins that share an identical genetic background. However, this is not the case, suggesting that while genes are an important factor in determining susceptibility to autism, environmental factors interact with those genes to really determine the ultimate risk. Another conclusion or observation from the genetic studies that supports a role for environmental factors is the fact that most genes that have been associated with ASD are not major effect genes. In other words, they themselves, in and of themselves, are not tightly associated with autism or don't cause autism. But rather, many of these genes seem to create modest vulnerabilities. In some cases, there are genes that have been shown to create major vulnerabilities, but even in genetic syndromes highly associated with autism spectrum disorders, for example, Fragile X, a significant percentage of the carriers do not have ASD. So this would again suggest that while genes are important, they're not the only determinant of risk for ASD. There's also evidence of de novo gene mutations that can increase risk for ASD. Most de novo gene mutations are reflective of an environmental exposure. So it's the environmental exposure that is driving the de novo gene mutation. And then the last piece of evidence from the genetic studies are that there are a number of gene variants that have been linked, I should say, to autism that confer altered vulnerability to environmental stressors and environmental exposures. So for example, genes that regulate redox or methylation patterns, genes that regulate heavy metal metabolism, and genes that are important in the metabolism of organophosphorus pesticides have been linked to increased risk for ASD. So again, establishing a potential tie between environmental factors and risk for ASD. Sort of linked to this is the fact that there's a great deal of clinical heterogeneity in ASD. So we see different severities of ASD, we see different comorbidities associated with ASD. And so if it were just genes alone that were driving this, we would not necessarily expect this. But if we have genes that are influenced by environmental factors, which can vary dramatically across individuals, this could explain some of the clinical heterogeneity that is associated with ASD. Linked to this clinical heterogeneity is the fact that there is now solid evidence of systemic and CNS pathophysiology in patients diagnosed with ASD. So for example, there's now evidence of oxidative stress, there's evidence of immune dysfunction, including neuroinflammation in the brain, and there's now evidence of mitochondrial dysfunction in individuals with autism. What's interesting about that in the context of the discussion about environmental factors and their role in autism is that many of these pathophysiological outcomes are known to be exacerbated by environmental factors, including air pollution, organophosphorus pesticides, and heavy metals. So do we have any evidence that there are specific environmental factors that do alter risk for ASD? There are 4 that I think are pretty well supported by experimental evidence and epidemiological evidence in the human population. One of these includes rubella infection during the first trimester of pregnancy. In utero exposure to thalidomide or valproic acid has also been linked pretty tightly to an increased risk of autism. Paternal age is another one. But I think the big question mark in many people's minds is environmental chemicals. There's certainly been a lot of discussion about environmental chemicals and risk factors for autism, but the issue is that efforts to date to identify specific environmental risk factors have produced a number of candidates, but few definitive hits. So some of the candidates that have been proposed in a number of scientific venues include heavy metals, such as lead and methylmercury, pesticides, which include organophosphorus pesticides, as well as organochlorine pesticides, and then the persistent organic pollutants, or POPs. The categories of chemicals that fit into the POPs include polychlorinated biphenyls, or PCBs, polybrominated diphenyl ethers, or PBDs, which are the flame retardants, and polycyclic aromatic hydrocarbons, or PAHs, which are typically found in air pollution and cigarette smoke, for example. So the question really has become, do we have solid evidence to tie any of these or chemicals beyond these groups that could really increase the risk of autism? So one of the problems in trying to identify or really get good experimental evidence that any specific environmental chemical may be a risk factor for autism is really understanding how these environmental factors might influence ASD risk. I think the model that's out there that many people are actively exploring is represented in this slide, and basically what this model is demonstrating is that genetic susceptibility interacts with environmental factors, and this is going to be influenced by the timing of the exposure to the environmental factor And these three interactions will drive ASD risk, severity, but also treatment outcome. So one of the things that I think is important to remember with respect to this particular paradigm is that there's a number of ways in which genetic susceptibility may interact with environmental factors. And so that's where the science is really focused right now, is trying to determine how it is that a gene that increases risk for ASD can be influenced by environmental factors, or vice versa, how environmental factors can be influenced by genetic susceptibility to really drive the clinical outcomes that we associate with ASD. So one of the challenges in really trying to identify specific environmental factors is if we really have this interplay between genes and environment. And one of the things I want to say right up front is that I think very few people believe that environmental factors in and of themselves per se drive autism or cause autism. Rather, they're really interacting with this genetic background to determine the risk for autism. But if this is really what's happening, then it creates a very difficult problem in terms of identifying specific environmental factors using epidemiological approaches. And that's illustrated in this slide here. So if we look at the first panel, we're looking at an autism-related deficit as a percent in the population as a function of the concentration or dose, if you will, of the environmental factor or toxicant. And so in a typical population, you'll have a bell-shaped curve, which basically illustrates that in any population, there will be individuals that are hypersensitive to the toxicant, so they will start manifesting symptoms of very low doses or concentrations of the environmental factor. And in the same population, we will also have a number of individuals who are resistant to the adverse effects of the environmental factor. So it takes a much higher concentration of that particular environmental factor to cause the adverse effect. Typically, what determines somebody as being hypersensitive or resistant is their genetic background. So they may not have very good DNA repair enzymes, or their genes may encode for an enzyme that increases the toxification of an environmental factor, which would make them hypersensitive. Or if they're resistant, they may have a genetic factor that encodes for an enzyme that's very efficient at detoxifying a chemical, and so they're somewhat protected from this chemical relative to the rest of the population. So what happens if we layer then the autism population on top of the typical population? And that's shown here on panel B. So if indeed the genetic phenotype of autistic individuals makes them more susceptible to some of the adverse effects of environmental factors, we'd expect that the dose-response curve for the autistic population might be shifted to the left relative to the typically developing population. So basically what this graph is showing you is that the toxic dose at which 50% of the population will be affected is at a lower dose in the autistic population relative to the typically developing population. Moreover, we have a greater percentage of hypersensitive individuals who are going to react to much lower doses of the toxicant than we see in the typically developing population. So what makes this really complex in terms of epidemiological studies is that we now consider the fact that autism is not a single genetic disease, but rather a number of genes can contribute to autism. We have a very diverse genetic background against which these environmental factors could react. And so one of the ways that people are thinking about this clinically is we have autistic endophenotypes, different clinical manifestations of the disorder. And so what you can see represented here are three different curves that could represent three different populations of individuals with autism that have very distinct and different genetic backgrounds. And so their relative sensitivity to any given toxicant could be very different, which is going to set up different dose-response curves. So hopefully what this will impress upon you is that epidemiological approaches are going to have a very difficult time really pinning a specific environmental chemical to an increased risk in autism if we have all these genetic backgrounds in the autistic population that are going to modify the way specific individuals will react to an environmental stressor. So we are taking an alternative approach in our quest for identifying specific environmental factors that are linked to autism. And our approach is not unique here at UC Davis. There are a number of research laboratories around the world that are using a similar approach. And that is to really understand what is known about the pathology of autism spectrum disorders, and then ask, can we identify environmental chemicals that cause similar types of pathological changes, either in an experimental animal model or in a human model. And so to really get at this handle, we have to sort of have a synthesis or an understanding of what is thought to be the case of the pathology that gives rise to autism spectrum disorders. And so I think very simply, there's really converging evidence and sort of a convergence of thought across the field that autism really reflects altered patterns of neuronal connectivity within the developing brain. And what do we mean by neuronal connectivity? And that's illustrated in the slide here. So connections or the flow of information within the brain is really determined by the neurons, which are the communicating cells in the brain. And neurons can be subdivided into three major compartments. There's the cell body, where the nucleus resides, and this is where all the DNA is also residing and gives rise to the proteins that are expressed in the cell. But neurons have a very characteristic shape, and that is that they extend two types of processes away from the cell body. These are divided into dendrites, which are illustrated here. Dendrites are the primary receptive surface of the neuron. Most of the information coming into this neuron will come in through the dendritic arbor, as it's referred to. So we can see here that there are inputs from these neurons in red. They're delivering information across this gap between the cells known as the synapse. That information is received here at the dendritic branch or the dendritic process of the neuron, and it flows down to the cell body. Once that information is collected in the cell body, a signal is going to be generated, and that signal will pass from this recipient cell down to the next cell in the circuit through the axon. So this is the second major type of process. So the axon is going to transmit information from the cell body to the next cell in the circuit. And here we see the next cell in the circuit here. So one of the critical things to take away from this picture is that the flow of information and the distribution of information in the brain is really going to be determined by the branching patterns of the dendrites and the axon in these developing neurons. So if there are more dendrites, I should say, in autism, that means there's going to be an increased receptive surface of the neuron, and it's going to bring more information into the cell. Conversely, if there's a reduction in the size of the dendritic arbor, less information can come into that particular cell. And so changes in either the number of axons or dendrites, the branching patterns of the axons or the dendrites, will change the way information is processed in the brain. The one thing I wanted to point out to you is that autism may involve altered neuronal connectivity, and that's the phrase that's used by neurobiologists to refer to how these neurons are connecting with each other, that this may occur not only in the brain, but also in the peripheral nervous system. For example, the autonomic nervous system and the sensory nervous system. The autonomic nervous system basically will control the GI system and the immune system, so this might contribute to some of those comorbidities that we see in patients with ASD. And of course, the sensory nervous system is going to transmit information about sensation to the brain and then back down to the peripheral sensory organs. And there is evidence that both autonomic and sensory nervous system connectivity may be altered in patients with ASD. So in order to really develop models for looking at how environmental factors may interact with genes associated with autism to change neuronal connectivity, we have to have a pretty good understanding of what are the processes occurring in the developing brain that really determine how these neurons are going to connect with each other. And so there are 6 processes that are generally agreed upon by developmental neurobiologists to be important determinants of neuronal connectivity. These include neuronal migration, so the migration of the neurons from their birthplace in the developing brain to their final resting spot in the mature brain. The development of interneurons, these are the neurons that really are going to modulate signaling between the long projection neurons that connect between different regions of the brain. Changes in neuronal program cell death. This is a naturally occurring process during the development of the nervous system in which an extra number of neurons are generated, and these are pruned back to the appropriate number during development. And these neurons are pruned through the process of program cell death. In addition, we have axonal growth and branching that I've already referred to, dendritic growth, but also plasticity. Plasticity refers to the fact that the structure of the dendrites can actually change in response to stimuli that are coming into the brain. And then synaptogenesis and synaptic plasticity. So synaptogenesis is the formation of synapses between two neurons in a circuit. So looking across these 6 neurodevelopmental processes that are the major contributors or determinants of how neurons connect in the developing brain, it's becoming more and more evident that autism in particular reflects alterations in the last 3. So these are later stages of brain development. So axonal growth and branching, dendritic growth and plasticity, and synaptogenesis and synaptic plasticity are thought to be the neurodevelopmental processes that are most impacted during the pathogenesis of ASD. The evidence in support of this include genetic evidence. So many of the genes that have been implicated as risk factors for autism spectrum disorders are modulating these last 3 processes, axonal and dendritic growth and the formation of synapses. In addition, if you look at the histology, if you will, of the brain in patients that have autism, there's evidence that these last 3 processes are perturbed in the brains of patients diagnosed with ASD. Okay, so that's the background. Now I want to get into really talking about the focus of today's talk, which is there evidence that organophosphorus pesticides may be risk factors for ASD? Many studies have come out calling for investigations into OPs as ASD risk factors, and so is there any experimental evidence that indeed they may be involved? One thing to note about the OPs is that these are still the most widely used pesticides worldwide, so here in the United States, but also in other industrialized countries and developing countries. And these are used not only in agricultural settings, as you see illustrated in this slide. So here's a crop duster that's applying pesticides to a field. Here are individuals out in a field spraying using backpack sprayers to control pesticides in a cotton field. But they're also used in suburban settings and in urban settings, primarily to control cockroaches. Cockroaches are thought to be a huge antigen that triggers asthma, which is also increasing in prevalence in the United States and other countries. And so OPs are typically sprayed in schools and residences to control cockroaches, to control asthma. So there's a lot of exposure to these particular class of pesticides. So is there any epidemiological evidence to support OPs as ASD risk factors? We actually have a large number of studies now, so reporting behavioral and cognitive problems in school-age children following chronic exposure to low-level OPs. What you see here on the right is one of these studies, a relatively straightforward and elegant study in which an investigator was studying Mexican children that worked, or I should say, children in Mexico that lived either in the foothills or the valley of an agricultural area. The individuals who lived down in the valley tended to have a higher pesticide exposure. These were the agricultural workers who were actually involved in spraying the pesticides and were exposed to crops that had been sprayed with pesticides, whereas individuals that lived in the foothills were not as closely related to the agricultural activities that were occurring in this region. And so the investigator asked 4-year-olds and 5-year-olds to draw pictures of people. And what you can see very clearly here is that children that were in the foothills region that had the lower OP exposure drew much more complex pictures than those children who lived in the valley areas. And this basically was taken as an indication that exposure to these pesticides during early development interfered with the behavioral maturation of these children. Interestingly, of the number of studies that have been published really looking at links between exposure to OPs and behavioral maturation or cognitive development in children, actually linked perinatal OP exposure to ASD. And this is a paper that was published by Brenda Eskenazi in Environmental Health Perspectives. Other studies have shown that susceptibility to ASD is influenced by polymorphisms or genetic variances in PON1. PON1 is a key enzyme involved in OP detoxification. And I'll come back and talk about these studies in a little bit in more detail. But anyway, so we have a pretty good wealth of information in the epidemiological literature really suggesting or making a link between exposure of the developing brain, human brain, to OPs and problems in behavior and cognition. And as I said, a couple of these have specifically looked at ASD as an outcome of exposure to these and have found a link. So what do we know about the mechanisms of toxicity? Well, it turns out we know a lot about the toxicity of OPs, primarily in the context of acute toxicity. These compounds are developed to target the nervous system of insects, but the molecular target in insects is also expressed in humans. And that molecular target is the enzyme acetylcholinesterase, which is illustrated here. So acetylcholinesterase functions in the brain to terminate signaling by the neurotransmitter acetylcholine. So you see acetylcholine is represented here. It binds to the acetylcholinesterase enzyme at the active site, illustrated here. When that happens, acetylcholinesterase then hydrolyzes acetylcholine or breaks it down to give rise to acetate and to CoA. These two breakdown products are then taken back up into the neuron and resynthesized into acetylcholine for further use as a neurotransmitter. So the important point here is acetylcholinesterase functions in the normal brain to terminate signaling by the neurotransmitter acetylcholine. Now, and you see that represented here, I should say. So here's acetylcholine, that's the abbreviation for acetylcholine. Acetylcholinesterase breaks that down into choline and acetate. When OPs are on board, they inhibit that enzyme acetylcholinesterase. So they actually bind to this active site and prevent the normal substrate, the normal neurotransmitter, from binding to that active site. As a result of the inhibition of acetylcholinesterase, there's a significant increase in acetylcholine that's present in the synapse. And this leads to an overstimulation of the downstream receptors, the receptors on the postsynaptic side or the cell that's being stimulated that are going to respond to acetylcholine. Over time, this overstimulation will actually cause a decrease in the number of cholinergic receptors. So we first have an overstimulation of cholinergic signaling in the brain that eventually resolves into a downregulation or decreased cholinergic signaling in the brain. So this is a very well-established mechanism of action that is responsible for the symptoms that we see in individuals who are poisoned by or acutely intoxicated by OPs. Now, it is known that there are disturbances in cholinergic signaling in at least a number of ASD patients. However, it's highly unlikely that this particular mechanism of action explains that. And that is because we very rarely see individuals in the United States presenting with acute OP intoxication, which results in seizures and it results in respiratory distress. And we can measure this relatively easily in the clinic by measuring the level of activity of acetylcholinesterase in the blood, which is a very good biomarker of the level of acetylcholinesterase activity in the brain. So while we know a lot about the acute toxicity of OPs, this is not likely to be an explanation for how OPs might contribute to ASD. So as indicated here, most mothers and children are not exposed to opiate levels that cause acute intoxication. So this really leads to the question of whether or not exposure to OPs at lower environmental doses that do not cause inhibition of acetylcholinesterase may actually interfere with neuronal connectivity in the developing brain. Again, because we think that altered patterns of neuronal connectivity may be responsible for autism. And so this is a question that a number of people have been actively investigating in experimental settings. So one hypothesis that has been put out there is that OPs may interact with Reelin. Reelin is a serine protease that is an important signaling molecule in the developing brain. And this is a very complex slide. The point of showing you this is that Reelin is an extracellular protein. It binds to receptors in the membranes of neurons, triggers a number of signaling cascades that basically result in changes in the cytoskeleton that control neuronal migration, and they also control dendrite formation. So they're controlling those neurodevelopmental processes that are important determinants of neuronal connectivity. The other thing that's really important to note is that Reelin is one of the genes that has been implicated as an autism risk factor. So is it possible that OPs might interact with Reelin to change neuronal connectivity in the developing brain? So before we get into that, just to review some of the data that links mutations in the Reelin gene to ASD. So one of the things that many of you might know is there's a very strong linkage between chromosome 7q and ASD. Interestingly, Reelin is found within the 7q22 region. So it's in that hot spot of the genome that's been implicated in autism. The other thing that's very interesting is that there is a spontaneous mutation in mice that is called the Reelin mice that basically that basically decreases the expression of Reelin in these particular mice. And this is, again, due to a spontaneous deletion of the gene. A number of people have compared the phenotype in the Reelin mice to what we know about autistic patients. And so if you look down through this particular table, you can see that in a number of brain regions, including the cerebral cortex, the cerebellar cortex, etc., that the types of changes that have been documented in the autistic patients also seem to occur, or there's something very similar that occurs in the brains of the Reelin mice. So these data collectively have given rise to the theory that changes in or mutations in Reelin may be important in autism spectrum disorders. So what happens with these mutations in Reelin that have been linked to ASD? The critical finding is that there are reduced levels of Reelin in the brain and serum of autistic patients. This is due to the fact that these mutations that have been linked to ASD typically have greater than 12 GGC repeats in the 5' untranslated region of the Reelin gene. So what does this basically mean? So the UTR of the Reelin gene basically controls the transcription or expression of that Reelin gene. And when there are greater than 12 of these GGC repeats, that's going to decrease Reelin gene expression by 25 to 50%. So again, this mutation directly explains the observation of the reduced levels of Reelin in both the brain and serum of autistic patients. Now these long, as they're called, Reelin variants, long because they have an increased number of repeats of this GGC region in the gene for Reelin, these have been associated with ASD in an initial study and in 3 follow-up independent samples. Interestingly, they were not found in 4 other independent samples. So out of 8 studies that have been done looking at an association between the long Reelin variants, 4 found an association with an increased risk for autism, whereas 4 did not. It's been proposed that the discrepancy between these studies may be due to genetic heterogeneity between the populations that were examined. Certainly a very realistic possibility, very plausible possibility. However, an alternative hypothesis that's been put forth to explain this discrepancy is that the manifestation of this genetic mutation in Reelin may have been influenced by exposure to OPs. So when individuals went back and looked at these 8 specific experiments, or I should say studies that were done, they found that the 4 where they found an association were in populations that were likely to have had a much higher exposure to OPs than the populations where an association was not found. So does this make any sense? Do we have experimental data to back that hypothesis up that OP exposure may influence the manifestation of these Reelin mutations? So as shown on this slide, there actually is some interesting experimental data to suggest there could be an interaction. So this is a study that was done using a mouse model, and chlorpyrifos, which is a type of OP, was administered to these mice from postnatal day 1 to postnatal day 6. This is a developing brain of a mouse. And the investigators looked at how this exposure to the OP influenced Reelin expression in the brain. You can see here we're looking at the full change in Reelin messenger RNA in both females and males. So looking across this, both the low and the high dose of chlorpyrifos significantly decreased the expression of Reelin in both females and males. And there appeared to be maybe a more enhanced effect in the males relative to the females. Interestingly, another study, independent of this one, looked at DFP, a second type of OP, and saw that it inhibited the activity of Reelin. So we have 2 pieces of experimental evidence to suggest that OP exposure in the developing brain could indeed interact with this Reelin mutation associated with autism, with both the OPs and the mutation decreasing the expression of Reelin. So this is a figure that was actually published by 2 investigators in Europe, Persico and Bergeron, in 2006. There's been a number of studies coming out to try and validate this, and there's been some validation of it. But the model they proposed is the following, is that the Reelin mutation itself is probably not sufficient to cause autism, that developmental OP exposure itself is not sufficient to cause autism, but that these may interact and that they may be further modulated by a second gene variant tied to autism, which is that enzyme PON1. I had previously, in an earlier slide, indicated that PON1 is an enzyme that's very important in the detoxification of OPs. So if you have high PON1 activity levels, you will detoxify OPs much more effectively than if you have low PON1 activity, in which case the toxic form of the OP is going to stick around a lot longer. So what are we looking at here? This is a little bit of a complicated slide. So the first thing to note is that there are two lines that are shown here, one in green and one in red. The green line represents individuals who have the typical variation of Reelin. So they have 7 to 10 of those GGC repeats, so this we consider normal or typical. Whereas the red line refers to individuals that have that expanded repeat, so they have greater than 12 GGC repeats in the 5' untranslated region of the Reelin gene. So to remind you, those that have this expanded GGC repeat are going to have a decreased expression of Reelin. So we look here on the graph, we see Reelin protease activity represented on the y-axis. And so if you look just within this group right here, you can see that there's decreased protease activity or decreased activity of Reelin in the individuals with that high number of repeats in the UTR region of the Reelin gene. Again, remember that blunts Reelin expression, so that makes sense. So now, let's look at what happens when we take these individuals and expose them to OPs. So remember that, again, OPs are thought to decrease Reelin expression. So upon this hit with OP, which is represented by the thunderbolt here, we have a further decrease in the expression of Reelin in both populations. Now, the interesting and important part of this graph is this red dotted line. This basically is the threshold for, in this case, correct neuronal migration, but it could also be the threshold for correct dendritic arborization. When the individual's Reelin protease activity falls below this threshold, there will then be abnormalities in neurodevelopment, and these abnormalities may be relevant to autism. So we can see here that in these individuals, there is no decrease below the threshold, and that's because in this group of individuals, they have high PON1 activity. So even though they have these other deficits that are going to decrease their Reelin production or their Reelin expression, that because they have high PON1 activity, the influence of the OPs is minimized. So they're quickly able to detoxify the OP exposure. Now let's look over here on the right. These are individuals that have low PON1 activity. So they're not going to be able to detoxify the OPs as effectively. Therefore, the OP will stick around in its toxic form for much longer in that individual and will have a bigger impact on Reelin production. So in this case, we actually see that the individuals represented by the pink line will fall below that threshold. So again, these are the individuals that have the Reelin mutation that's associated with ASD, and under normal conditions without OPs, they're safe. But then when they're hit with the OPs, because they also have low PON1 activity, that drives their Reelin expression below this threshold. So again, this is simply a model to explain how we might have these interactions occurring between genetic susceptibilities and environmental factors that are going to influence the risk for autism. And as I said, there's been a lot of experimental research going on to try and verify or validate this model, and that work is still ongoing. Is that the only hypothesis to explain how these OPs may influence ASD risk? And the answer is no, that there are some alternative hypotheses out there. And one of these relates to the fact that acetylcholinesterase, which is the primary molecular target for OPs and acute OP intoxication, serves a second function. While its primary function in the adult brain is to break down that neurotransmitter acetylcholine, in the developing brain, acetylcholinesterase also functions as a morphogen that promotes axon outgrowth. So when acetylcholinesterase is present, axons are going to grow faster, and this can be used to basically target the axon towards the downstream neuron in the circuit that's being established. So the hypothesis that my lab has been investigating for the last couple of years is that these OPs may disrupt axonal growth by interfering with the morphogenic activity of acetylcholinesterase. So the particular OP that we've used for our studies is chlorpyrifos, which you'll see abbreviated as CPF throughout the rest of the slides. This is a phosphorothionate pesticide. Might be important to some of you, but it's a type of OP that is used for crop protection, and most importantly in the context, I think, of autism, it's also used in flea dips and roach control sprays. So here's the parent compound, chlorpyrifos. The parent compound in and of itself is not particularly toxic, or thought not to be particularly neurotoxic. It is metabolized by an enzyme known as P450 to give rise to the oxone metabolite, or CPFO. Basically what this does is it removes the sulfur group here and replaces it with an oxygen. This is important because this now becomes a phosphate, and it can donate or phosphorylate target molecules in the developing brain. CPFO is the toxic entity. It can be detoxified, as we previously discussed, by the enzyme PON1, and this gives rise to the presumably non-toxic metabolite known as TCP. So what we know about chlorpyrifos is it is a developmental neurotoxicant. There's also documented widespread exposure of children not only in agricultural settings but also in suburban and urban populations to this particular OP. In fact, some of the data that's out there suggests that urine samples from a significant percentage, anywhere from 40 to 100% of children in the U.S. contain these metabolites of CPF. There's also an NHANES 2 report that indicates that the level of CPF metabolites in children are twice those found in adults. And then a number of animal studies have shown that the developing brain is much more sensitive to the neurotoxic effects of chlorpyrifos than is the mature brain. So what happens when we put chlorpyrifos into our experimental model systems? And the experimental model system that we're using are cultured neurons from the sensory system. So these are DRG, or dorsal root ganglion neurons, and we've exposed them either to vehicle or to vehicle containing chlorpyrifos. And this is just an image of these neurons after 24 hours in culture, showing what's happening to their axon outgrowth. And so if you look here in the top panel, and these happen to be neurons that were derived from wild-type animals that expressed acetylcholinesterase, you can see that relative to the vehicle, chlorpyrifos seems to inhibit axon outgrowth. So it's actually, the axons are shorter in those cultures that have been exposed to the chlorpyrifos. Now what's very interesting about this is that if acetylcholinesterase is the molecular target that the chlorpyrifos is interacting with to decrease axon outgrowth, then if we remove the acetylcholinesterase, we should see no effect of chlorpyrifos. And that's what's illustrated in the bottom two panels here. So these are DRG neurons that were derived from mice that have been genetically engineered to no longer express acetylcholinesterase. So there's no acetylcholinesterase in the system. You can see that in the neurons from these ACHE nullozygous mice, or the mice not expressing ACHE, that their neurons are shorter than the wild-type neurons in the presence of vehicle. This basically validates that acetylcholinesterase is important in axon outgrowth. Now what's really interesting is if you look at the neuron derived from the acetylcholinesterase knockout mouse in the presence of chlorpyrifos, you can see that relative to the vehicle control, there doesn't seem to be much impact on axon outgrowth. The axons are about the same size. What's really interesting is the axon length is pretty similar to the axon length in the wild-type neuron that was exposed to CPF. So this suggests that CPF is inhibiting axon outgrowth through its interactions with acetylcholinesterase. You need acetylcholinesterase there in order for the chlorpyrifos to have an effect on axon outgrowth. These data were quantified, and the quantification of those data are shown in this particular slide. And so what you're looking at here is total axon length per neuron, and this is measured in micrometers, as a function of exposure to chlorpyrifos, so the various concentrations of chlorpyrifos. On the left-hand side of the slide are the neurons that were derived from the wild-type mice that expressed acetylcholinesterase, and you can see, as illustrated in the pictures previously, that relative to the vehicle control, which is the white bar, chlorpyrifos seems to inhibit axon outgrowth, and this happens in a concentration-dependent manner. Now, if we look over here on the right side of the graph, these are the data from the neurons that were derived from the ACHA knockout mice, and these don't express acetylcholinesterase. And you can see that in the vehicle control that there is a decrease in axon outgrowth relative to the vehicle control from the wild type. What's very interesting is that regardless of the concentration of chlorpyrifos to which these cultures are exposed, there's no decrease in axon outgrowth relative to the vehicle control from these knockout animals. So again, suggesting the acetylcholinesterase must be present for chlorpyrifos to cause axon inhibition. So that's pretty cool, but that's done in a cultured neuron. Do we see the same effect of chlorpyrifos in vivo? It's actually pretty difficult to measure axon outgrowth in the intact nervous system, and so we moved to an alternative model other than a rat or a mouse, and that was to zebrafish. And one of the reasons for this is that there are zebrafish out there that are engineered to express a green fluorescent protein in subsets of neurons. So we can actually track these axons throughout development very readily in a zebrafish. And so these are some data from these experiments. And you can see here that we're taking zebrafish larvae. So this is 0 would be the time of fertilization of the zebrafish eggs. And then we look at 24 hours post-fertilization. I'm sorry, let me back up there. So we actually expose these zebrafish to chlorpyrifos oxone from 24 hours post-fertilization out to 72 hours post-fertilization, and then assess axons, the length of the axons at this time point. And so what you see in this figure here is basically the photographic evidence of the effects of chlorpyrifos oxone on axon outgrowth in this particular transgenic animal. Here's the midline of the animal represented by the dotted lines here, and then we're looking at the dorsal axon and the ventral axon in these particular animals. This is from vehicle control, so you can see what the normal patterns of axon outgrowth would be like in this particular zebrafish. This is what happens when you expose these zebrafish to CPF. We can start to see that there is a diminution or a decrease in the length of the axons of these particular animals, and it appears to be more prominent on the dorsal axon than on the ventral axon. These data are quantified over here. So we're looking at percent fish with intact dorsal axons in the upper left-hand graph here. And you can see that there's a decrease in that as a function of exposure to chlorpyrifos oxone. If we look at the percent dorsal segments that are innervated, or the percent of dorsal segments that have an axon, we can see that this is also decreased by exposure to chlorpyrifos oxone. Again, another measure of axon outgrowth is the length of the axons. This is also decreased by CPFO. We don't see quite as much effect on the ventral axon, and when we went back to the literature, we found out that the dorsal axon actually is more influenced by acetylcholinesterase than is the ventral. So this fits with the hypothesis that CPFO or CPF is interfering with axon outgrowth by interactions with acetylcholinesterase. So one of the things that's very interesting about this is that acetylcholinesterase is important not only in axon outgrowth, but it's also important in synaptogenesis in the developing brain. And so we actually went off and did a genome array, so we did a gene array experiment to look at what are the genes that are being altered by chlorpyrifos in the developing rat brain. As I'd indicated, it's pretty hard to look at axon outgrowth experimentally in the brain, but we can look at synaptogenesis by looking at the genes that are involved in synaptogenesis, and we did this in a rat brain. I'm not going to walk you through this in any detail because there's a lot of information in here, but the bottom line is that if you look across this information, you can see that those kinds of proteins or genes that are involved in neurotransmission and synaptogenesis or synaptic plasticity are significantly altered by exposure to chlorpyrifos. So it appears that chlorpyrifos may be altering neuronal connectivity directly by interfering with axon outgrowth, and then either indirectly or directly interfering with synaptic plasticity in the developing brain. So how could this chlorpyrifos exposure be changing synaptogenesis? One way might be by altering axon outgrowth, because I talked to you earlier that axon outgrowth determines neuronal connectivity, and if neuronal connectivity is altered in the developing brain, that will change synaptic transmission in the brain. Another way, though, is by actually interfering with molecules that drive synapse formation in the developing brain. And one of these molecules is neuroligand. So it turns out that neuroligand has a great deal of structural and functional homology with acetylcholinesterase, or ACHE. So here you see a representation of neuroligand across the top. And in this part right here, there's a lot of homology to acetylcholinesterase. So this is both at the structural level and, as I said, above at the functional level. There are studies in which it's been shown that the function of acetylcholinesterase can be replaced by neuroligand. Vice versa, if neuroligand is removed from a cell, some of the function of neuroligand can be replicated by expressing acetylcholinesterase. So they can swap out for each other functionally to some extent. As some of you may know, neuroligin has been linked to ASD. So mutations in neuroligin is one of the few familial gene mutations that has been directly linked to increased risk of ASD. Neuroligin works to cause synapse formation by binding to beta-neurexin. And so what you see in this figure over here is that beta-neurexin is expressed on the presynaptic side, so this would be the incoming axon, whereas neuroligin is expressed on the postsynaptic side, on the dendritic side. These two interact to basically stabilize synapses. There are 4 different types or isoforms of neuroligin, and these differentially will affect excitatory synapses versus inhibitory synapses. So the bottom line is these neuroligins are very important in stabilizing synapses in the developing brain and in modulating synaptic plasticity during signaling in the developing brain, and that mutations in neuroligin have been linked to ASD. And then finally, that there's a great deal of structural and functional homology between ACHE and neuroligins. So collectively, these data suggested that potentially these OPs may be interacting with neuroligin to change synaptic plasticity in the developing brain. We've done a couple of preliminary studies to investigate this hypothesis, and here we're showing that chlorpyrifos alters the balance of excitatory to inhibitory synapses in cultured hippocampal neurons. So we're looking at this ratio between excitatory and inhibitory synapses, and we can see that chlorpyrifos at a concentration of 1 micromolar increases that ratio. So it increases the number of excitatory receptors relative to the number of inhibitory receptors. This is actually pretty interesting because there is evidence that in at least some types of autism, there's an increase in excitation to inhibition in the brain. On the right-hand side, we have another graph looking at the effects of CPFO, and we can see a very similar effect of CPFO, although the concentrations are significantly lower, which is consistent with the fact that CPFO seems to be the toxic metabolite. So collectively, these data sort of raise the intriguing possibility that OP exposures during critical periods of development may amplify ASD-related gene mutations in neuroligin to disrupt synapse formation, and that this interaction could then alter patterns of neuronal activity associated with ASD. And as evidence that this might not be so far-fetched are some really exciting data that were published in 2012 in Proceedings of the National Academy of Sciences by Virginia Raw and her colleagues at Columbia University. And so they actually used imaging of children to look at the distances between different brain regions of children that were exposed to very low levels of CPF versus children that were exposed to relatively high levels of CPF. And I'm not going to walk you through the slide, number one, because I'm not an expert in imaging in the brain, and number two, it's pretty complicated to explain in the context of a webinar. But the bottom line is that there is a very clear correlation between exposure to CPF and the distance between different brain regions, different regions of the brain, I should say, in these children. And so the distances are sort of a proxy measure for neuronal connectivity. So this is basically telling us that there appears to be altered patterns of neuronal connectivity in the brains of children that have been exposed to CPF. So I just wanted to end by discussing what these findings mean to parents and to clinicians. So I think what's important to take away from this is that chemical exposure, both pre- and postnatally, can influence clinical outcome. And by that we mean the types and severity of the behaviors, the comorbidities, and perhaps even the response to treatment. While this is kind of scary, I think it's actually got a glimmer of hope to it in that we can actually more readily control chemical exposures than we can control genetic factors to prevent or mitigate the expression of ASD-related traits. And so while we don't yet have a really good handle on specific chemical exposures that are related to ASD, research that's moving forward to identify these, I think, is well worth the effort because, again, we can actually control exposures to these chemicals. So in the meantime, is there enough evidence to suggest that perhaps you want to control your exposures to chlorpyrifos and other OPs? I'd say absolutely yes. So how can you do this? So one way is to minimize or prevent your exposure to chemical contaminants during pregnancy and early childhood. Again, I want to reiterate that the evidence would suggest that it's not just prenatal exposure that's important, but early postnatal exposure may also be important. So several straightforward, easy ways to minimize or prevent your exposure. Number one, don't use OPs in the home or in either your home or in your yard. When you go to buy any kind of insecticide at the hardware store, know what you're getting. Know what kind of class of compound this is. There's a number of very useful guides that are available readily on the internet to identify any given chemical as an OP or other type of pesticide. The other thing is to consume organically grown produce if this is a possibility that's accessible to you. There are good studies showing that children who consume organically grown produce have significantly lower body burdens of OPs than children who consume typical produce. If you can't afford organic produce or it's not available to you, another very easy, straightforward step that you can take is to be sure to thoroughly wash all produce before serving it to your children. If it has a cuticle on it, remove the cuticle. So this means paring carrots, removing the peels of potatoes, etc. Also, you can work with your local agencies to minimize use of OPs in public places and or to get them to post notices to the public of OP spray schedules and locations so you can avoid those for a certain period of time after the exposure. And then last but not least, keep the dust levels in your homes as low as possible. It's been shown that these OPs tend to absorb to dust particles, and that homes that are dusty, that children in homes that are dusty have higher OP body burdens than children who live in homes that have lower dust levels. It's also important to note that these dust particles have been shown to accumulate in stuffed toys, so it's also a good idea to wash the stuffed toys routinely to remove that. And with that, I'd like to end my presentation and acknowledge the individuals who contributed to the research that I presented to you and the funding sources that provided the support for those studies to occur. And I am happy to take questions at this point.
So your questions are down in the chat log. Would you like me to read them to you, Dr. Lein, or do you want to read them and then answer them?
Whichever works for you. Would you like me to read them?
That would be great.
Okay. So the first question I see is, can you explain how failed dendritic arborization manifests both on a cellular level and in a potentially functional one? So on a cellular level, it's important to note that maybe not failed is the correct word, but abnormal dendritic arborization can either be too much dendrites or too little dendrites. As with any other biological process, appropriate or normal physiological function depends on a balance. And so we do know that dendrites typically grow in response to learning or in response to experience. However, pruning those dendrites are also a very important part of setting up functional neural circuits. So either too much pruning or not enough pruning, or too much growth or not enough growth can, on a cellular level, give rise to aberrant or differently functioning neural circuits. And so cellularly, that's what happens. And so this can lead to functional changes. For example, I think one of the best described of these is that in schizophrenia, there's now compelling evidence that much of the functional deficits associated with schizophrenia can be tracked back to inappropriate pruning of dendrites during adolescence. And so, again, I don't want you to think that it's either too much or too, let me say this, there's not a definitive pattern of dendritic arborization that will cause functional deficits, but it's a change from the normal balance that will cause or be related to functional deficits. And this has been shown in a number of animal models, and we now have experimental or clinical data from humans to suggest this is true as well. Okay. So next question I see is, I would like to know your opinion about detoxification therapy for autism disorder. Do you think it's an effective treatment method? I'm not a clinician, so that's number one. So I am a basic scientific researcher. So I go by what's in the literature. And based on my reading of the literature, I think there is not a lot of evidence to support detoxification therapies in general. That many of the therapies that are out there will remove not only the toxicant of concern, which is typically not at acutely intoxicating levels, but will remove a lot of other very important minerals, for example, that are essential for normal brain development. So I think it's unwise to plunge into these, that it really is a discussion that you need to have with your provider, your health provider, to determine if this is an appropriate action to take with your particular child. And this should be based on some pretty solid evidence of what body burdens are for the toxicant that you're trying to eliminate. So again, I would proceed with great caution. I don't think the evidence is really solid that these do a great deal of good in the majority of cases, and there's actually evidence that they can be harmful in at least some cases. So again, something to proceed with caution on, I think. Okay. Okay, one slide was not on the PowerPoint sent to us. The slide with the study with the 4- and 5-year-old drawing. Is this one you can share? Yes, I'll be sure to connect up with Denise Fulton and make sure that that gets put into the public domain so you have access to that. Our study is underway now to determine how GMOs and ASD increased respectively over the past 2 decades. That's a really good question. I'm not aware of anybody that is actively pursuing that. And for those of you that don't know, GMOs are genetically modified organisms. Certainly, GMOs have changed the pattern of pesticide use significantly, and the impact of that on autism at this point is sort of anybody's guess. So I think that's a wide open question, and I'm not aware of any data that specifically address that. Okay, can you speculate on what kind of behaviors or developments happen in PCB-induced arborization? You mentioned inhibition of inhibition or excitation of auditory sensitivity. Can any other areas be predicted, or is the arborization and its impact random? Okay, I think this is referring to a presentation I gave about a month ago on PCBs. I did not talk specifically about PCBs in this particular presentation, but so if I look back at this, I think that the effects of PCBs are not generally random, but they do impact a number of different regions in the brain. So we do know that PCBs will increase dendritic arborization in the hippocampus and in the cortex and also in the cerebellum. But what we haven't done is gone back and looked to see if there are regions of the brain where we don't see an impact of PCBs. We know the molecular target of PCBs is ryanodine receptors, so the prediction would be that neurons that express ryanodine receptors are going to be susceptible to the effects of PCBs, whereas neurons that are not expressing ryanodine receptors during exposure are not going to be susceptible to the effects. So whether different brain regions are more or less susceptible is a question that we're actively exploring now, and I don't have any data to give you on that. But I wouldn't think it's just generally random, but there is some specificity in the effect. Next question is, do you know of research on autism addressing the potential role of ionizing radiation? Another good question. I don't know of any research in that particular area. I know that there is research on ionizing radiation in the context of developmental neurotoxicity, but I'm not aware of any data specifically addressing autism, which doesn't mean it's not out there, but I'm not aware of it. So the next question is, example on how thalidomide and valproic acid, which ones and how they would be exposed in utero. Okay, so I'm not quite sure I understand that, but there is data specifically tying exposure in utero to thalidomide and valproic acid to increase risk for autism. What I could do is, actually I'll refer you to some reviews that kind of go over that literature if you'd be interested in that. I can send that to Denise offline and she can make that available hopefully to you. So paternal age, this is a study that was done by Dr. Hertz-Pichotto here at UC Davis, so you should be able to find it in the public press using her name as a search engine. But basically, it was increasing paternal age was tied to an increased risk of autism. And I will be honest and tell you I don't remember what specific age ranges were most highly correlated with autism, but certainly it was that as paternal age increased, there was an increased risk of autism. And again, that is a published study, so you should be able to pull that out of the peer-reviewed literature. So pesticides, examples please, ones used in agriculture and at home. So we talked about chlorpyrifos. This is one that has been phased out by EPA for use in the home, but it still actually can be applied by commercial applicators in a residential setting or in a school setting, for example. So chlorpyrifos you will find in urban and suburban and agricultural areas. Other examples of OP pesticides that you are likely to encounter include diazinon. This is commonly used to control ants. It is currently being phased out for purchased by the homeowner, but is still being used by commercial applicators. Other types of pesticides include malathion, and of course all of these can have a number of different commercial names. So there's a very good website out there that's, and I will get the exact web address to Denise if people are interested. But if you just go online and type examples of organophosphorus pesticides, you'll come up with a number of great websites that will give you a definition of what are OPs, where they can be found, and what their common trade names are. There's actually a pesticide registry in the state of Oregon, which has a great website. There's also a pesticide information hotline here at UC Davis. So there's a number of resources available on the net where you can, if you just, again, type in organophosphorus pesticide examples, you'll come up with a great list describing what these are, what the chemical names are, and how they're commonly used. Okay, POPs, examples in plain English, please. Okay, so persistent organic pollutants basically refers to environmental contaminants that are long-lived in the environment. And so these are diverse types of chemicals. Many of these are man-made chemicals, although some occur through natural processes such as combustion. So the three main categories of POPs that people talk about all the time include the polycyclic aromatic hydrocarbons, or PAHs. These are the types of chemicals that are formed during combustion of organic materials. So these are formed during cigarette smoking. They're formed whenever there's a forest fire. They're formed when you burn charcoal in your grill. And so these are the components that are found on our charred meats. An example would be benzanthracene, and if any of you have heard of that, or benzopyrene are sort of the two classics. But again, PAHs are formed during the combustion of organic materials, and these are a type of POPs because they're long-lived in the environment. Another example of the POPs that I talked about were the polychlorinated biphenyls, or PCBs. These are also man-made chemicals that are long-lived in the environment, and they were basically developed and synthesized by man because they're very stable chemicals. So they dissipate heat very well. They stand up to heating and cooling very well. So these are chemicals that were used in transformers, electrical transformers, to dissipate the heat that's formed in a number of applications that require electricity. They are also put into paints to stabilize paint so they wouldn't break down under high heat or low heat conditions. They're also put in insulation and caulking and stuff like that. So these are just, again, another category, another type of persistent organic pollutant, or POP. Dioxins also fall into that same sort of category. Many of you have probably heard of dioxins. And then the other category of POPs that I talked about were the PBDEs, polybrominated diphenyl ethers. These are the brominated flame retardants that many of you are probably familiar with. Okay, next question. PCB studies have been only done in the United States or other countries? I am from Costa Rica. My brother is Asperger, and my nephew is autistic. My son is autistic. We have always thought it was genetic. Would it be accurate to say that the exposure happens initially by the mother through the womb or breastfeeding? Okay, so there's a lot of questions buried in there. So PCB exposures have been documented in every country in which they've been examined. And that's because these PCBs are very long-lived in the environment, and they biomagnify up the food chain, so they've been dispersed throughout the world. So they're basically globally distributed. Excuse me. So they're found in very high concentrations in populations that tend to eat predator populations, so people who eat predator fish, The Eskimos have very high body concentrations because they eat polar bears. So basically, all of us have PCBs in us. There's been a number of studies that looked at human exposure, and every population in which it's been examined, PCB body burdens have been documented. They're higher again in some populations than others, primarily based on diet. So it is plausible and likely that there would be PCBs in Costa Rica. So the exposure depends somewhat on the class of chemical we're talking about, but I think it's absolutely fair to say that the exposure could be either through the womb, or it could be through breastfeeding, or it could be through consumption of contaminated products by the child themselves during early brain development. What's really important to keep track of or to know is that neuronal connectivity is being established in the brain, the developing brain, while the child is in utero, but also after birth. So there's a period of about 4 to 5 years where there's very active synaptogenesis going on in the brain. And this period of synaptogenesis and synaptic pruning will continue until up to adolescence. And so brain development in humans continues long after birth, and the primary aspect of brain development that occurs after birth is these patterns of neuronal connectivity. And so I think that the opportunities for exposure that could influence risk for autism are certainly in utero during brain development, but also postnatally. So there's a number of different ways, a number of different exposures that could be very important in determining risk for autism. It's also important to note, however, that when the exposure occurs, is likely to give rise to different outcomes. And that's because different regions of the brain are developing during different periods of development, and that there are different neurodevelopmental processes that are occurring at different stages of neurodevelopment. So a very complicated story, which is another reason why it's been very difficult to identify specific exposures that are going to give rise to increased risk for autism. Okay, so next question. I watched your earlier presentation on June 1st, wanted to ask, in the PCB study, it does seem strange that the low-dose group showed a stronger difference than the high-dose group. How many dams were used to reduce the low-dose set of rats? And did you look at differences by dam to see if babies from one dam may have been contributing strongly to the effects seen at the low dose? Okay, so this is a really good question. Why is it that we see more or greater effects, amplified effects at the low dose relative to the high dose of PCBs? So again, this is not relevant to the talk I gave today, but to the presentation I gave on June 1st. So we don't really know the explanation for this sort of reverse dose response curve that we reported. We're actively investigating a number of hypotheses to explain that. But the bottom line is that we don't think it's because we had an aberrant response in one of the low-dose dams. The number of dams we had in the low-dose group and the high-dose group were identical. So we had 12 dams in each group, and we only took one male and one female pup from each of those litters to run the behavioral assays. So we actually had 12 females and 12 males in the low-dose group, 12 females and 12 males in the high-dose group. Each of those came from a different dam. So we don't think that the response that we're seeing is being driven by the fact that we just had one dam that was giving us a very abnormal effect. Okay, I think I answered everything from that question. Another question on PCBs. Why would lower levels of PCBs have a more dramatic effect? I'm not sure I follow. That is a question, as I said, that we're actively exploring. It may have to do with effects on calcium levels in the cell. As you may remember from my presentation on PCBs, we have tied the effects of PCBs on dendritic arborization to increase calcium in cells exposed to PCBs. There's some literature out there in the developmental neurobiology field saying that the response to increased calcium will very much depend on where the endogenous levels of calcium are at the time of the increase. So in other words, if at the time of the exposure to PCBs, the cell has a relatively low level of intracellular calcium, the increase in calcium caused by the PCBs will enhance dendritic arborization. However, if the cell has a relatively high intracellular level of calcium at the time of exposure to the PCBs, then further increases in calcium can actually cause dendrite retraction. So in the low to mid range of calcium, we get increases in dendritic arborization. In the mid to high levels of intracellular calcium, we actually get dendritic retraction, and probably because the calcium is triggering different signaling pathways internally. So at low levels of PCB, we're in that low to mid range of calcium, we'll get dendritic arborization, increased dendritic arborization. At high levels of intracellular calcium, when PCBs come on board, pushing the calcium level even higher, that will lead to dendrite retraction. So that's one of the hypotheses that we're actually investigating right now. Okay. So next question. It seems like the mutation decreases Reelin in production and organophosphate exposure compounds this. Yes. Is there a way to test for this mutation? In the absence of the mutation, can high exposure manifest the same way? I suspect that there are ways to test for the mutation. The mutation is pretty well described. I don't know that it's a commonly used genetic test. I doubt it, but with the right resources, I'm sure that can be tested for. And I think this next question of whether a high enough exposure to OPs could cause the same effect is a really good one, and one that it's really, we don't know the answer to. And the reason it's difficult to kind of answer this is because when we get at high enough levels of OP exposure, we then start to get into acute OP intoxication. We start to inhibit the enzymatic activity of acetylcholinesterase, which is going to change cholinergic signaling in the developing brain. And how that might affect Reelin expression is not known. So I think it's an experiment that needs to be run, and I don't know that anybody's actually run it yet. Because we're usually not interested in very high concentrations of OP because we know that most individuals are not exposed to these really high concentrations of OPs. Why do we know that? Because if they would be manifesting with seizures, they'd be manifesting with respiratory distress, with muscle tremors, all the classic signs of acute OP intoxication. And clearly we don't see that in the majority of the mothers and children that are at risk for autism. So that seems to be unlikely. Okay, next question. Is there any way theoretically to upregulate the production of Reelin? That is a really good question too, and theoretically, yes, there are ways to upregulate Reelin. Whether any of these are practical is another question, clinically practical. So it's been known, for example, that BDNF, or bovine-derived neurotrophic factor, which is a very important factor in neurodevelopment as well, an endogenous growth factor, that this can increase levels of Reelin expression. But to my knowledge, there's been no successful clinical applications of using BDNF to drive any kind of activity in the brain. People have tried for other applications, primarily for Parkinson's disease, but to date there's not been much luck in that. Next question, is Reelin's activity unique or like many detox functions duplicated by other cells or chemicals? So in the developing brain, there is a lot of redundancy in signals that drive dendritic outgrowth, for example, or that drive neuronal migration. However, each of the factors that are important in controlling these are activating relatively unique signaling pathways. So I think it's fair to say that Reelin, which is an endogenous compound, this is not a toxic compound, it's basically a natural gene product in the developing brain. It's very important in normal neurodevelopment. That Reelin expression basically is relatively unique, although there are other factors that work with Reelin to really determine the final migratory pathway of any given neuron or that control dendritic arborization in any given neuron.
So Dr. Lein, you can skip the next couple you've already answered, but there are a few more down at the bottom.
Okay. Okay, here's one that looks unique. You mentioned the spraying of pesticides for protecting crops, but what about the pesticide or chemical DDT that's used to control the spread of mosquitoes? Please explain, if you can, if this is bad and could cause ASD or cancer in the brain once consumed. So DDT is a different class of pesticide. I've been talking about the organophosphorus pesticides. DDT is one of the organochlorine pesticides. Basically, the OPs were developed to replace the organochlorines because the organochlorines are actually very persistent in the environment. Organophosphorus pesticides tend to break down relatively quickly in the presence of sunlight or in the presence of water. So there have been 2 epidemiological studies of which I'm aware that have made a link between exposure to organochlorine pesticides, not specifically DDT, but a group of organochlorine pesticides, and increased risk of some types of ASD. So, for example, pervasive developmental disorder. I'm not aware of any experimental studies that have tried to figure out a mechanism by which DDT may contribute to increased risk of autism. But since DDT and other types of organochlorine compounds are affecting neurotransmission as well, it's not inconceivable that they might interfere with neuronal connectivity. But again, I'm not aware of any studies that have specifically looked at this, and none that have looked at it in the conjunction of a genetic mutation that's been associated with autism. So the next question is, it seems that some children with ASD are more severely affected with extensive stereotypy and apraxia despite intense intervention, personal observation. Is there an affected area in the brain or associated with a specific neurotransmitter, like I've read dopamine associated with motor control and GABA with language? I'm going to have to punt on this one. This is outside my immediate area of expertise, and again, I'm not a clinician. I do know that there are a number of studies going on that are trying to link various neurochemicals, so neurotransmitters and other molecules that are involved in neurotransmission, to specific aspects of autism. So, for example, there's a lot of interest in looking at glutamatergic and GABAergic signaling in the context of seizures associated with a subset of patients with ASD. But with respect to stereotypy and apraxia, I really don't know. And again, this is just the fact that this is not my area of expertise, so I'm going to have to defer an answer on that particular question. Okay, explain how living near a dump or landfill with a lot of toxicity could affect a child to have them to be diagnosed as ASD. So there are actually several papers that have come out now and been published, epidemiological studies, that have shown an association between living next to a landfill or living next to a polluted area and increased incidence in autism. And so it's thought that, again, as we discussed earlier, that it's the environmental chemicals that are toxic chemicals associated with these landfills that are leaching either into the water table, or it could be volatilizing or in the airspace that the children living adjacent to these sites might be exposed to. So again, the hypothesis is that because of the proximity to the landfill or the hazardous land site, that these children have a higher body burden of a number of toxic chemicals. The question to be resolved, of course, is what are the specific toxic chemicals that may be linked to an increased risk of autism. So again, I think there is evidence out there that living near a polluted area could be associated with an increased risk of autism, but the specific factors that are responsible for that have yet to be determined.